All right, in this video, Unit 9, Mendelian Genetics, looking at 9.5 applications in genetics. So this is like things you can do with genetics. We're also going to be looking at a little bit of human genome just because it's needed a place to be, and this is the place for it to be. All right, so selective breeding. This has been going on as long as there have been people. And so selective breeding is when humans take traits that we desire in organisms, animals, plants, whatever, and we breed those traits into particular organisms. Take dogs, for instance. Dogs came from wolves, or domestic dogs came from wolves. We took traits that we liked in wolves, and particularly like tame wolves, and bred those traits, and then eventually we came up with dogs that are completely useless, like those little ones. But then some of our big dogs are still useful. But what if you take this too far? Well, that's what's called inbreeding. So just so we understand, most pure-blooded animals or dogs are inbred dogs, right? Because they've taken traits that they like in dogs and they've bred them into other dogs. And so this causes what's called inbreeding, right? Usually like if you see on this particular graph, what's happened, you have two snakes, they've got siblings, and then two of those siblings had some more offspring. And notice what happens. That deleterious allele is what's called it there. So, deleterious, to look at the word there, delete. It's a bad thing, right? And so it can come back out because of that inbreeding. You see this in animal populations and human populations sometimes, and it's never good. It's never a good thing. But inbreeding is necessary to get traits like what you see in like dog breeds. And so that's why a lot of dog breeds have some problems. Genetic engineering. This is when you have more what you'd think of something done in a lab, right? So this is when changes are made to the genetic code of an organism from the outside. Genes are inserted, genes are manipulated some way. We're going to look at lots of examples of genetic engineering. And so pretty much everything we're getting ready to talk about is going to be under that heading, including transgenic organisms. Transgenic organism is an organism that has had its genes changed from some genetic application, inserting a gene into it that can cause that organism to have certain types of traits, right? You see this with animals. You can see this with plants. Particularly when we talk about our plants, especially those plants that we eat, we call them something else, right? We call them GMO foods. GMO is just genetically modified organism. GMO food is the food that has been genetically modified. Why would we genetically modify it? To make it more nutritious, to make it more appetizing, to make it more appealing, all these different things, right? Or to make it easier to grow with a lot of crops that we have: corn, cotton, soybeans, pretty much any crop that you can think of that's going to be one of the major crops in our country or in this region is going to be somehow genetically modified. There's not really much out there that hasn't been. As far as our foods are concerned, it's going to be some foods are genetically modified, some are not. In general, this is just changes in the A's, T's, C's, and G's. And so there's not been any studies that have done to prove that genetically modified organisms or genetically modified foods in particular are unhealthy for us to eat, or that they cause particular allergens or any of those things. There's just no real proof for that. So there's that. So let's look at a couple ways that viruses can be used. One of those is vaccines. So here's a example that's more recent, I guess, with the COVID vaccine. And a virus is basically used in order to gain the genetic code, and that genetic code is injected into an organism. The organism makes a protein, and then that protein elicits an immune response. In some other vaccines, portions of a virus were used in order to inject those portions of a virus into an organism, or into us in that case, in order to elicit an immune response. And it's basically to prepare us for the actual disease, right? When you come in contact with it, your immune system will have already been encountered it. Now, that's the issue with some vaccines are kind of hit or miss, because the virus is going to be mutating so quickly that you kind of have to guess. COVID vaccine is a good example of this now. Flu vaccine is another good example of that. And so, whereas some viruses are going to mutate more slowly, and so a vaccine is very good. Like chickenpox is a great example of this. Smallpox, they're able to eradicate diseases completely because of vaccines, which is good. Gene therapy, also using viruses. And so this is where you take a gene, the desired gene, or we'll say the correct gene, and make a virus that is able to bring that gene, and then you give that to someone who has an incorrect gene, meaning that the gene is mutated and is creating an incorrect phenotype, like a disease or something like that, and you insert that into them, and supposedly it will correct their cells. This could work with cancer cells. This could work with other things that are more like more systemic, cystic fibrosis, something like that. There's a lot of tests being done with gene therapy. It's kind of one of the new medical marvels of the future. Kind of cool. Cloning. It's exactly what it sounds like. It is making an identical organism. Now, what cloning is not, it is not taking an adult sheep and like photocopying it to where you have another adult sheep. When you clone an organism, you're actually creating an identical egg, basically, or an identical zygote. And you implant that zygote into a surrogate mother, and then that mother is pregnant and gives birth to an identical organism. And so that's when we're talking about cloning, that's what's going on. The sheep, of course, in 96 was the most famous cloned animal. Since then, people have their pets cloned. It's kind of interesting. And they pay a lot of money for it. And their pets have like glow-in-the-dark noses and feet just to show that they're like cloned. Look it up. It's really cool. So yeah, interesting. Stem cell research. This is where we take stem cells. Stem cells are cells that are undifferentiated, right? They could be completely undifferentiated, meaning that they're really like an embryonic stem cell that is really early in development, or they could be later in development, like a skin stem cell or a muscle stem cell or a bone stem cell, and basically reprogramming those cells so that they can then regrow muscle tissue or nerve tissue or bone tissue or whatever sort of tissue. And that's the idea. Similar to gene therapy in that regard, with not using a virus, of course, just reprogramming a cell to be able to like undifferentiate itself, to regrow things that have since become regulated to not grow anymore. And so stem cell research, very important medical ideas as well. So let's look at the human genome real quick. This is kind of switching gears. Human genome contains 46 chromosomes or 23 pairs of homologous chromosomes. There's a picture of those chromosomes. It's called a karyotype. And then 22 of those pairs are what we call autosomes. Auto means self, remember that? An autosome is just a regular chromosome, an everyday chromosome. And then one of those pairs, the 23rd pair, is going to be a sex chromosome, either XX in females or XY in males, which we're going to talk about. Actually, we did talk about that with sex-linked traits already. So you can see the XY chromosome, or the Y chromosome, is a lot smaller than the X chromosome. And the X chromosome is necessary for life. The Y chromosome is not necessary for life. Couple of traits, or there's some examples of traits that are not inherited. And so let's look at that. So height is an example. Height is inherited. You do inherit whether or not you're going to be tall from your parents, right? But human height has increased over the last 150 years. Why is that? Well, it's because we have grocery stores. That's the big change. Nutrition is an example of something that can change height because we're eating better than we were, right? And so eating better than we were is not genetics, right? It is just allowing those genetics to do their thing, as opposed to, someone who's not eating good is not going to grow well. And so that's the whole idea. Intelligence is another one. You're born with a capacity for intelligence, but if you're never exposed to things, if you're never taught to learn, if you're never challenged to learn, you're never going to really find that potential, right? And so there's several of those that you could look at that are traits that we have as humans that are not necessarily inherited. A lot of times you'll hear them called environmental traits. Sometimes you'll hear the phrase nature versus nurture, and so there's some several traits that you could talk about that are not inherited but are influenced by environment, or kind of a mix of the two. Let's look at blood types. Remember, blood types are codominant. A and B are both dominant, and if you have them together, they both express. O is recessive. Nothing to really add to that, other than When you have type A blood, you have anti-B antigens in your blood. So if you came in contact with B, you would fight it, right? That's why like a blood transfusion is really important. Well, someone who has AB doesn't have any of those antigens, right? And so they can accept A or B. Well, someone who has type O is actually against A and B, and so they can only accept type O blood, right? But just so happens that type O is the most common blood type, and AB is actually the rarest blood type, and so you have that kind of interchange there, which is interesting. And you've probably heard of like A negative, B negative, O positive. Where does that come from? Well, it's this thing called the rhesus factor. You don't need to know anything other than the rhesus factor is just another protein that's found on blood cells. And if you're Rh positive, you have it. If you're Rh negative, you don't have it. Really doesn't cause much issues. Could cause an issue with someone who's pregnant and their child is Rh positive and they're Rh negative. And it could cause issues there, but we have medicines for that. It's pretty normal. Let's talk lastly about human pedigrees. Pedigree is just a way to show like a family tree, but then to track a particular condition over the course of that family tree. In pedigrees, the squares are males, the circles are females, and the colored in shape, like you can see there in generation 1, that square is colored in. That will denote the condition that is being tracked. And so in this particular instance, notice that the condition is dominant. How do you know? Well, the dad in generation 1 has the condition. He has the dominant allele. And it's really easy to see that. Everyone who's homozygous recessive does not have the allele. Those who have the dominant allele have the condition. Well, let's look at another pedigree. What about this one? A lot of times pedigrees are going to be numbered like this to help you kind of say, well, how does individual 6 have the disorder if neither of the parents do? It's because the parents are both heterozygous, right? They're able to both pass that recessive allele. There's a 25% chance that they'd give that to their offspring, and that's exactly what happens here. They give that to their offspring, and then their offspring have, well, this is a really interesting family tree. I just noticed that. Oh well. Sometimes things, yeah, whatever. So anyway, you can track that just by using that kind of just basic Punnett square. Usually just a monohybrid cross will allow you to be able to track that.